Hello everyone. Today I'm going to talk about a small molecule inhibitor from a Chinese medicinal herb, Salvia miltiorrhiza, screening of it against 3-cell main protease of SARS-CoV-2. SARS-CoV-2 has 4 structural proteins: spike, nucleocapsid, and a membrane and envelope. And as its genomic material, it has RNA, which is 30,000 nucleotide long, and it can code for 29 viral proteins, and including spike. Along with this, 16 out of these 29 viral proteins are known as non-structural proteins, or NSP, and 7 out of these 16 are enzymatic in nature. Today I'm going to focus on one of the important enzyme of this virus, which is NSP5. NSP5 is also known as 3-cell MPRO or 3-cell main protease. Here you can show a bimole representation of this enzyme, and this green color represent active site, which is a catalytic diode, and the deep colors represent a dimer interface from these 2 subunits. This enzyme is a homodimeric cysteine protease, which helps the virus to get its 11 out of 16 non-structural proteins into mature form, including itself. So, and according to the literature, this monomer of this enzyme is almost inactive. So, inhibiting this enzyme via active site inhibitor or taking down the dimer interface can lead to the viral inactivity. So, in the lab, I expressed this recombinant enzyme, and I did characterization to find out the optimal working condition for this enzyme. As you can see, the kinetics has been done here. The optimal pH is around 7.5. I checked the effect of salt, and NaCl is kind of detrimental for this enzyme with increasing concentration of it. After finding optimal working condition for this enzyme, I screened 31 natural tanshinones against this enzyme, and I got 2 leading compounds, TO6 and T29. After that, I did the IC50 using these 2 inhibitors, and I got the IC50 value for both of them less than 20 macromolar. How these things are doing against the real virus, to find out, we did a cell assay where, as you can see, the TO6 expected inhibit the virus as well. But T29, which is, work really good in the in vitro assay, but it didn't do very good in the cell assay. Although we have new thing which is unexpected T30, which didn't do well in the in vitro assay, but did very well in the cell assay, which indicates there are some alternative target for this inhibitor as well. To get a hint where these inhibitors are binding, I did the docking as well using molecular docking method using Autodock Vena, and as you can see, except for this one, most of the bindings are at the dimer interface, so which kind of indicates that this inhibitor may be acting as a dimer interface and taking down this dimer form. In conclusion, from this 31, I got 2 leading compounds, and molecular docking indicates the distal binding of it, and TO6 in the cell show IC50 of 20 macromolar, and other inhibitors shows a good activity, which indicates that it has alternate targets. For future work, I will screen these inhibitors again from the 3CL from various variants of SARS-CoV-2. I will try to understand how exactly these things are working, inhibiting this enzyme, and I will try to screen with other enzymes as well to find out the other targets for this enzyme, for this inhibitor, sorry. I want to thank my lab, and Dr. Brome, Dr. Yasinta Batabai, Dr. Preeti Panwar, Jimin, and Stephen for the enormous help on my work. Thank you very much, everyone.
Questions?
That was really interesting. Great talk.
Thank you very much.
What kind of cells is it inhibiting?
in Kaku cell line.
Which one? Kako?
Kaku cell line.
So, is there any kind of application that you can think of then that might be steering toward a certain function? Can you further investigate the specific function of those cells that is being inhibited?
Sorry, will you please repeat once again?
Can you further investigate what specific function is being inhibited in those cells? And then maybe exploit that inhibition mechanism.
Yeah, that could be. At this moment, I do not have the answer for that. But yeah, maybe. But we tried in others previously with this inhibitor using cathepsins in the cell line, and there was no other inhibition was there except for the cathepsin K, and specifically not the. So it's just breaking down the active site, breaking down the oligomerize rather than breaking down the stopping the active site. So by that way, this is kind of leaving everything unaltered, rather inhibiting everything, that specific enzyme altering its structure or its activity or inhibiting other stuff. So that's why we are trying to figure out how these inhibitors are working with this enzyme. So if it's working in the same way, taking away the dimer interface and leaving the active site unaltered for it, then it will serve our purpose.
Thank you.